This particular figure gives us a good sense of how meiosis can produce genetic diversity, assuming genetic diversity is there to begin with. So in this particular example, we're looking at a hypothetical cell, and 2n in this particular cell would equal to what number? And hopefully your answer was 2. 2n is equal to 2. There's one pair of homologous chromosomes. Now, you'll remember on these chromosomes are loci, which are genes. So we're saying the B gene is located on this end, the A gene is located on this end. You'll notice that there are two different alleles for each gene, the dominant allele and the recessive allele. So we have a dominant and recessive for the A, we have a dominant and a recessive for the B. So the genotype for this particular individual would be dominant A, recessive A, dominant B, recessive B, or in other words, a double heterozygote. Big A, big B, little b would be the genotype. Now it's important to remember that when meiosis takes place, each gamete must get one allele from each gene. In other words, each gamete has to have one A and it has to have one B. And we can see that's the case on the far side. Now, if we have truly independent assortment coupled with crossing over, then that means each possible package of alleles in a gamete has an equal probability. So if you look at it at the beginning, if I can get one allele from each gene, there are 4 different ways this could happen. I could get a dominant A with a recessive B. I could get a dominant A with a dominant B if crossing over occurred. I could get a recessive A with a dominant B, or I could get a recessive A with a recessive B, again, if crossing over occurred. So when meiosis begins, our DNA duplicates. So now we have 2 sister chromatids, and it would make sense that the allele content on the chromatids is the same, because we said when the DNA replicates, it replicates to produce an identical sister chromatid. If in meiosis 1 crossing over occurs, then what that means is I now create a situation where I have the potential for genetic diversity. In other words, the chromosome from dad, let's say, had the dominant A, the recessive B. The one from mom always had the recessive A with the dominant B. But if you look over at this situation, excuse me, you can see we have the type from one parent, dominant A, recessive B, the type from the other parent, recessive A, dominant B, but I've now got new combinations. I've got recessive A with recessive B, and I don't see that anywhere over here. I have dominant A with dominant B, and again, I don't see that anywhere over here. So what crossing over has done is resulted in genetic recombination. In other words, I've recombined or put the alleles together in a new way that wasn't observed in either parent independently. So what happens in meiosis 1 is homologous chromosome split. What happens in meiosis 2 is sister chromatid split. So I produce 4 types of gametes. Now, this gamete is just like a parental. This gamete is just like a parental. So the first and last gametes are said to be non-recombinant, or parental types. The 2 middle gametes have a unique combination of alleles that wasn't observed in either parent individually. Again, I don't see a little a with a little b, or I don't see a little a with a little b. So what I have is a situation for the potential of genetic diversity. Now, I just noticed in this particular figure they made a misprint here. So the little a, little b is fine, but we should have this gamete being dominant A and dominant B, because that would be the second type of gamete produced when the sister chromatids separated. So please make that correction. Again, I just noticed it in the course of discussing this figure with you. So I would produce 4 types of gametes. 2 of them would be parental types, 2 of them would be non-parental. Again, this one would be recessive A, recessive B. This one should be dominant A, dominant B. So I have 2 recombinants or non-parentals, 2 non-recombinants or parentals, and each would occur in equal frequency. In other words, I would have 25% of this type, of this type, of this type, and of this type. And that's going to be important when we talk about Punnett squares and inheritance, which is going to be the topic in the next question.